It is my honor and pleasure to introduce our two speakers for our accredited session entitled The Promise and Potential of CAR T Cell Therapy in Rheumatology. And we have, again, this sort of collaborative intersection between oncology and rheumatology. And our speakers today are doctors David L. Porter and Peter Merkel. And I will introduce both of them and then welcome them to the stage, and they will do their presentations in their entirety. And we will then, as per usual, moderate a Q&A session. So make sure to, again, put your questions through the app so that we've collected them throughout the program. And at the very end, we will try to get your questions answered by our illustrious panel here today. So our first speaker is Dr. David L. Porter. He is the Jody Fisher Horowitz Professor of Leukemia Care Excellence at the Perelman School of Medicine and Abramson Cancer Center. He is the Executive Director of the Center for Cell Therapy and Transplant at the University of Pennsylvania. He chairs and serves on numerous local, national, and international committees focused on hematologic malignancies, stem cell transplantation, and cell therapy. Dr. Porter has been the primary or co-author of over 200 research and reviews, and along with colleagues at the University of Pennsylvania, he has pioneered the successful development of CAR T cells for cancer. Our second speaker is none other than our Real Men Wear Purple t-shirt wearing, making it popular, making it hip, Dr. Peter Merkel. He is the chief of rheumatology and a professor of medicine and epidemiology at the University of Pennsylvania. He is an internationally recognized research and clinical expert in vasculitis, scleroderma, and other systemic autoimmune diseases, and he is author on over 400 scientific publications. He's the principal investigator of the Vasculitis Clinical Research Consortium and the Vasculitis Patient-Powered Research Network. These are two leading international research infrastructures for clinical investigation in vasculitis. His research focuses on development of novel therapies for systemic autoimmune diseases, clinical trial design and conduct, outcome measure development, clinical epidemiology, genetic epidemiology, and if that wasn't enough, biomarker discovery. He has received the majority of his research support from the NIH, the US FDA, the Patient-Centered Outcomes Research Institute, with additional funding from industry sources and private foundations. Again, it is my honor and pleasure to introduce our two speakers, Doctors Peter Merkel and David Porter. Please welcome and come on to the platform.
Well, thank you. I say hi to fellow members of AWARE, as I am a proud member, and we welcome Dr. David Porter, who I've been working on steadily the last several years to transform him into a rheumatologist. He's close. He's an honorary rheumatologist. I think we wanted to, this is our title slide. We're going to do this together. Overall, it's the title up there, and then we have individual sections. You've already heard about Dr. Porter's disclosures. He does a lot of work to develop things, as do I. I actually think these do matter. We are very involved in helping to develop drugs for our patients. Today, we are going to introduce CAR T therapy in history and development oncology. It is an incredible story that Dave is going to tell you about. I mean, it's truly a thrilling story of how this came about. We're going to talk about the rationale for the use of CAR T and other cell therapies for systemic autoimmune diseases. We'll talk about the importance of the close collaboration between oncology and rheumatology to study and administer CAR T therapy safely and scientifically accurately, and we'll take some questions and answers. So that's our goal for today. We've worked together on this, and I will hand over and go blank to Dr. David Porter.
Well, thank you all. Thank you, Peter, and thank you for that kind introduction. I think, yes, I don't have a purple T-shirt yet. I am anxiously going to look forward to getting one. I do appreciate the invitation to be here. I think just the fact that I was invited highlights that our fields are intersecting. Oncology and rheumatology have been tangential, I think, specialties, but now are really tightly intersecting. And I really appreciate the opportunity to be here. You'll see how certainly at Penn, rheumatology and oncology and cell therapy really are now working tightly together. Just the fact that Dr. Merkel and I are doing this together. My task on this My task on this really is to give you a little bit of the background about CAR T cells. What are they? Why are they so exciting? And really try and highlight the potential. And then I'll turn it over to Dr. Merkel to talk to you a little bit more about the potential in autoimmune disease. I'm going to start out just by reminding people of things that I'm sure everybody knows, but that most of the cells in our body, and certainly immune cells, cancer cells, all have very well-defined fingerprints. They have surface proteins that can serve as targets for new therapies. Cancer and immune cells have these well-characterized proteins. We know that they can be targeted with monoclonal antibodies or engineered antibodies, immune cells. The things I'm going to highlight mostly are targets on the surface of B cells. I'm going to tell you mostly about work with CD19, but it could be BCMA, it could be CD20. You really can pick your target. We do work with CD22. And we can target these antigens on the surface of B cells. The first one really that got developed was CD19, because CD19 on some level is a really ideal tumor target. Its expression is really limited to B cells and B cell precursors. Though CD19 is also expressed by just about every B cell malignancy. And we, in my field, in oncology, feel that B cells are expendable tissue, if you will. We know that patients can live without B cells. We too use a lot of anti-B cell therapies, whether it be chemotherapy or monoclonal antibodies. And we have learned, as have the rheumatologists over years, how to manage patients with very low or with no B cells. And we are targeting these with chimeric antigen receptors. And I apologize taking you back to Greek mythology on a Saturday afternoon. This may give people sort of the cold shivers just going back to high school or college. But the term chimeric antigen receptor, the chimeric, really comes from the chimera, that mythical Greek beast, the head of a lion, the tail of a serpent, the body of a goat. And what it really means is something made up of different parts. And a chimeric antigen receptor is made up of different parts. It's a molecule that on the outside has an antibody moiety that can target whatever target you want. I'm going to show you how this targets CD19 in the initial cases in cancer. In the middle is a hinge region that allows this molecule to be stably integrated into the T cell membrane. And a lot of the work is done by the internal fragments. These are the co-stimulatory domains. And in most successful cases has been the CD3 zeta signaling domain. So when the antibody recognizes its target, it activates the T cell through CD3 signaling and co-stimulation. Either the most traditional ones have been 4-1BB or the CD28 co-stimulatory domain. And so that's the anatomy of a CAR molecule. The way that this works is really cartooned here. If you understand this, you understand CAR T cell therapy. We use gene transfer technology, in our case at Penn, or in most cases, this is through viral vectors bringing in new genetic material, either a lentiviral vector or a retroviral vector, bringing in this new genetic material into the T cell. The gene gets stably integrated into the genome, conferring novel antigen specificity. So we use this lentiviral vector. In our case, it brings in a gene for the chimeric antigen receptor molecule. It's stably integrated into the genome. It directs expression of the chimeric antigen receptor, which gets transported and integrated into the cell membrane. And it now allows this T cell to recognize its target, CD19 in this case, when it couldn't before, leaving behind, hopefully, a dead tumor cell, where you could replace that and just say a dead B cell. Clinically, the way it works is shown here. Everything in blue happens in the patient and everything in yellow happens in the lab. Patient comes into the office, they undergo a steady state leukapheresis to have their T cells collected. Those T cells are brought to the lab down in the lower left corner. I'm guessing I'm using my pointer, and I'm guessing you're not able to see that. But number two here, the cells are infected with this virus, lentiviral vector or retroviral vector or other mechanisms to get new genetic material in them. In three, the cells are expanded in the laboratory until they're enough for clinical use. And then they're brought back to the patient up in the upper right-hand corner. Patients typically receive a lymphodepletion or immunosuppressive chemotherapy regimen. The idea here is to lymphodeplete their own native T cells to make room for the new incoming T cells to allow for things like homeostatic proliferation and expansion of the new T cells that they're getting. The cells are brought to the patient. They are infused just intravenously. This can happen as an inpatient or an outpatient, depending on the patient, depending on the product, and actually the infrastructure of the center where this is done. Historically, this process has taken about 7 to 14 days to grow these T cells in the lab. There are novel manufacturing methods now where this can be done in 1 or 2 days, and the cells can actually be ready quite quickly. So why all the excitement? Why is CAR T cells such an exciting field? Why did this really make so much news? And I think the answer is really here. When CAR T cells were first developed, and we treated our patient back in 2000, our first patient back in 2010, they were tested in patients with highly refractory, multiply relapsed B-cell malignancies, relapsed refractory ALL, lymphoma, CLL, multiple myeloma. And all of these patients, particularly in the initial trials, had almost no probability of remission or cure. And in fact, the vast majority of these patients would be destined to die of their malignancy within a few weeks, if not a few months. And yet, despite the fact that most of these patients would have died without effective therapy and had very few options, you can see here in the right-hand column that the complete response rates vary anywhere from about 50 to 100%, and long-term leukemia-free or disease-free survival anywhere between about 30 to 75% in these patients. And in fact, we now know that many of these remissions have been sustained for long periods of time, going beyond 10 years, going out to 14 years, again, in a patient population that was dying of their disease. And I would venture to say that many of these patients have likely been cured of their refractory malignancies. There are now, after these first initial studies, there are 7 FDA-approved CAR T-cell products for 7 different indications. All of these indications have in common that they are B-cell malignancies, and you can just see the different products here. And in fact, there are also 5 cell therapies that are non-CAR T-cells FDA-approved. I want to give you a little bit of sense of the magnitude of the effects that these CAR T cells can have by giving you 2 examples and showing you some outcomes that are expected in patients who have relapsed refractory ALL. While ALL is really an incredible success story, now patients, up to 90% of patients, particularly pediatric patients with ALL, can be cured with standard chemotherapy. But when it relapses and becomes refractory, the prognosis is dismal. The median survival is less than a year. Less than a quarter of these patients will survive beyond 3 years. While we do allogeneic stem cell transplant for many patients with ALL, it's largely ineffective when patients have refractory disease to the point that we usually don't offer it. And there has been this desperate need for more effective therapies for these patients who develop refractory leukemia. The first reported study was here in the New England Journal of Medicine. Tisagenlecleucel is the name of the product manufactured by Novartis under the name Kymriah. This was a clinical trial led by colleagues of mine at Children's Hospital of Philadelphia, Shannon Maud and Steve Grupp, treating pediatric patients with tisagenlecleucel or Tisa-cel CAR T cells. And really the outcomes that were quite astonishing. There were 75 patients in this trial. Again, these patients had relapsed refractory disease. They were vast majority destined to die. 81% had a complete remission. Minimal residual disease testing at a level of 1 in a million was negative. And you can see in the left-hand panel here the overall survival and event-free survival. And you can see there seems to be a very nice plateau in the event-free survival curve, and this has held out over many years, where about 50% of these patients have likely been cured of their disease. This was the first study in pediatric ALL, and this is what led to the first FDA approval of a genetically modified T cell. We called it CTL-019 at the University of Pennsylvania. It was licensed to Novartis and marketed under the name Tisagen Lifileucel or Tisacel in August of 2017. We had done these initial studies, and what this led to at Penn was this flash mob. This was a picture that was taken in our atrium the day that these CAR T cells were FDA approved, and I show this slide for several reasons. This was August 30th, 2017, almost 7 years to the day that the first adult patient with CLL was treated, and about 5 years since the first pediatric patient was treated. That was really lightning speed, considering this was FDA approval of a genetically modified human immune cell. But the other point of this slide is look at all the people. These are people who all had some connection to development of this therapy. All of these people were somehow involved in development of CAR T cells, either in the clinical trial, the manufacturing, the correlative analyses. And it was really quite an enormous effort. Director of our cancer center, Bob Vonderheide, is right there in the middle, standing with the microphone in his hand. Again, highlighting the magnitude, what I'm showing you here on the left side is the probability of event-free survival in these patients with ALL with conventional standard therapy. And as I told you, their prognosis is dismal. Very, very few patients survive beyond a year, let alone 5 years. Event-free survival is about 10%. Yet CAR T cells have led to this curve that I showed you with event-free and long-term survivals of about 50%. So that is the magnitude that we see with CAR T cells in ALL. Similarly, right after this, CAR T cells have been applied to non-Hodgkin's lymphoma, diffuse large B-cell lymphoma initially. We know that non-Hodgkin's lymphoma can be curable with standard types of chemotherapy, perhaps in maybe 60% of cases. But of course, and it's always the but, again, patients with relapsed and refractory lymphoma have a terrible prognosis. Those refractory to chemotherapy or who relapse within about a year of, say, an autologous stem cell transplant have anticipated very low response rates to their next line of therapy. The likelihood of responding is about 20 to 36%. Only about 8% achieve a complete remission. Similarly, patients who fail a second line, what we call salvage therapy for relapse, have a terrible prognosis. And in these groups of patients, there has also been this urgent need for better therapy. This is the anticipated outcome for patients with relapsed refractory large cell lymphoma. You can see the long-term survival is about 20% of patients. We clearly have to do better. CAR T cells then were next applied to patients with relapsed or refractory diffuse large B-cell lymphoma who have this terrible prognosis, again, in a study led in part at the University of Pennsylvania by my colleague Steve Schuster and published in the New England Journal of Medicine here. Just some high-level data. There were 93 patients, 52% responded, 40% had a complete remission. The ongoing response rates perhaps were not as exciting as we saw in ALL. But again, considering that most of these patients had no options and were likely to die, long-term remission rates were about 30% and now about 40% with other products as well. And this just shows you graphically some of these outcomes: duration of response, progression-free survival, survival, et cetera. And the point that I want to make with this slide is really one thing. Patients still relapse, but they tend to relapse early. The vast majority of patients who fail CAR T cells have progression within the first 3 and maybe the first 6 months. And if you look, after about 6 months, there is very little subsequent relapse. And that is important. So rather than talking about things like median survival and median progression-free survival, we really look at sustained outcomes, sustained progression-free and overall survival over time. And I highlight that because, again, these patients were destined to relapse quickly, and yet they are now living not just the 1 and 2 years that you see here, but beyond 5 and 10 and 15 years. There have now been 3 different CAR T cells FDA approved for diffuse large cell lymphoma. I show them here. I want to highlight that this is a side-by-side presentation. This is not a comparative trial. It is very, very difficult to compare across clinical trials. We all say that. We all show a similar table where they are listed side by side. It's hard not to compare, but they are different trials, different patient populations. But if you look at the 3 different CAR T cell products, you can see it's fairly consistent across different CAR T cells. Overall responses in 50 to 80% of patients, complete remissions in 40 to 55% of patients, and importantly, the long-term ongoing responses in about 30 to 45% of patients. So these responses really are quite dramatic. That said, there is very unique and specific toxicity that makes this a very makes this field really very specialized because of its unique toxicities. There's really no infusional toxicity. As I mentioned, we can do this often in an outpatient setting. There is a risk of tumor lysis syndrome when you kill all the, large numbers of cancer cells all at the same time. Some people refer to that as a toxicity. That, in my mind, is actually a testament to the potency of the therapy. But the really unique toxicities are what's referred to as the cytokine release syndrome and a very unique neurologic toxicity. And I will highlight also that when effective, since normal B cells have CD19, many of these patients develop B cell aplasia and long-term hypogammaglobulinemia. Again, is that a toxicity or is that a sign of efficacy? The cytokine release syndrome correlates with CAR T cell activation and expansion in the body. We know these cells can expand about 10,000-fold in the body. It always starts with a fever by definition that happens about 1 to 14 days after the infusion, but these fevers can be quite dramatic, up to 105, 106 degrees. It's associated with pretty severe flu-like syndrome, like myalgias, arthralgias, anorexia, nausea, diarrhea, and it can escalate to things like capillary leak, leading to hypotension and hypoxia. In many cases, it's self-limited, though we now know that direct anti-cytokine therapy can be dramatically effective for the cytokine release syndrome. There is mortality related to this, but it has gotten much safer over time. In part because we very early on learned the mechanisms. It was seen that interleukin-6 is markedly elevated in patients with cytokine release syndrome, and at the same time seeing this dramatic immune activation with HLH-like biochemical parameters, dramatically high ferritins and CRP, et cetera. Because IL-6 was felt to be central and at very high levels, one of these very first patients who had severe CRS was treated with tocilizumab. Tocilizumab is an IL-6 receptor antagonist. It blocks IL-6 mediated effects in rheumatology. It's a disease that you all, or a drug that you all were probably way more familiar with than the oncologists. And it can rapidly reverse the signs and symptoms of CRS. We also know that earlier treatment, and this third bullet point, with tocilizumab doesn't impair the activity of the CAR T cells, and so we can treat these patients early and prevent some of the more severe side effects. CRS-related mortality was initially reported in the early days of this field at about 10%. I will tell you now it's in the low single-digit range, somewhere between about 1 to 3%. Just showing you here the kind of dramatic impact tocilizumab has had on this. In this left-hand panel, you can see the patient who developed CRS at about 6 days after receiving CAR T cells, with this dramatic rise of ferritin showing immune activation up to about 600,000. At this point, this patient was becoming hemodynamically unstable. And in the right-hand panel, you can see his fever curve, 105, 105.4. Here's where he's hemodynamically unstable and gets tocilizumab, and you can see this rapid drop, normalization of his fever within a few hours, and the immune activation signs markedly improved as you can see his ferritin rapidly going down. The other very unique toxicity is this neurologic toxicity. It's referred to in the field as ICANS, for immune effector cell associated neurologic syndrome. It's a terrible acronym, and I apologize, but it's really caught on to call this ICANS. The mechanisms for the neurologic toxicity are less well understood, and the management, because of that, is less well defined. We do know that the onset usually is within the first 2 to 3 weeks of CAR T cell infusion, and it almost always follows development of the cytokine release syndrome. In many cases, it is self-limited, though in rare cases, it can progress to seizures, cerebral edema, and even death in very few cases. But it also has some very unique features. It generally presents with an encephalopathy, with delirium, lethargy, headaches. There is this unique dysgraphia, which I'm just showing you an example of on the right-hand panel, such that we monitor this by asking patients to write the same sentence on a piece of paper every day. You can see this patient here 4 days after their infusion writes their I love Shawnee, KS. The next day, while they're having this neurologic toxicity, their handwriting is unreadable. This was self-limited in this case, and by the next day, they can write again normally. So in addition to dysgraphia, they may get aphasia, memory impairment, again, can progress to things like focal neurologic deficits, seizures, even coma, hemorrhage, cerebral edema, and death in the more severe cases. But again, in general, we see very few severe cases these days. Typically happens after the cytokine release syndrome. We know that tocilizumab is not effective. IL-6 is probably not central. We manage these patients really based on their clinical symptoms with supportive care. They get anti-seizure medications. Corticosteroids are the first-line therapy, and there's now some very interesting data that anakinra, an IL-1 receptor antagonist, may in fact be effective, at least in some cases, with this neurologic toxicity. This is one of the reasons, in part, that we ask patients to remain near the treatment site for the first 28 days after their infusion, because of the risk of developing neurologic toxicity a little bit later than the CRS. But this has indeed been a success story. Once people began to understand the mechanisms of the toxicity and toxicity management, this has become much safer over the last 14 years. The house staff at the University of Pennsylvania in the early days of CAR T cells coined the term catastrophe because of how sick these patients get. But again, as I wanted to highlight, the treatment-related mortality initially maybe 10% is now in the low single-digit range, but it is still significant, and I think really not acceptable until that number is 0. And that's really what we can expect in using CAR T cells for malignancies. And I can show you similar examples in other B-cell malignancies. But I think what's really exciting is what next? Of course, everybody wants a new CAR, right? And so what are the CARs of the future? Where are we going with this? This is what we refer to as kind of the universe or solar system of CAR T cells. What this is showing you is all the CAR T cell products in development. And in fact, this is just the CAR T cells in development for oncology. The green are the autologous products being developed. The red are the allogeneic products being developed. Those in the middle of this universe in the dark gray are those products that are marketed. As you get further out toward the rim, these are products further out in clinical development. So around the rim here are those products in very early preclinical development. As they get into clinical trials or in animal models and then clinical trials, they move more and more toward the center. So this field is expanding rapidly with dramatic numbers of new CAR T cells. In fact, if you just look at our institution, we have a CAR T cell trial for just about every malignancy, not just B cell malignancies, AML, lung cancer, pancreatic cancer, et cetera. You can just read the list here. In the solid tumors, it's been more difficult to see responses. Occasionally, there will be responses in some of these non-B cell malignancies. There have been very few long-term good responses to date, but there is progress, and it's set steady, though it has been slower than what we see in B cell malignancies. So clearly, CAR T cells have altered this treatment paradigm for B cell malignancies, but really, while there's a lot of work in solid tumors, the outcomes haven't yet been dramatic. And I think the really next exciting frontier is applying CAR T cells beyond cancer, which is why I think everybody is here today and listening to this. This is just the beginning, of course. I showed you that universe slide. These are a listing of all the different biotech and pharma companies that are developing cell therapy products to treat autoimmunity. This is just a rapidly expanding field. You can see just about, every major manufacturer that you can think of has a program in developing cell therapy for autoimmunity. And if this indeed comes to pass, if these are successful for patients with autoimmune disease, B-cell malignancies, solid tumors, we really have the potential for a large traffic jam with all these new cars being developed. But I think I want to just end this part about the application of CAR T cells to B cell malignancies by highlighting one thing that I have kind of hinted at as I've been talking, and that is using the word cure. Are we curing these patients with these awful leukemias and lymphomas, et cetera? I don't know how many oncologists you all interact with, but you may know that oncologists don't like to use the word cure. We're all superstitious. We say things like disease-free survival. And well, we know that when we give CAR T cells to these patients who have had highly refractory relapse disease, many of these patients have minimal residual disease negative responses at a level of about 1 in a million cells. We can't find any residual cancer. We also know that in the majority of these cases, the relapses are anticipated to happen very early, and most of these relapses in all these clinical trials happen in that first 3 to 6 or maybe 6 to 12 months. Yet we have patients alive in remission well beyond 12 months, now beyond 10 and out to 14 years. So we still say things like CAR T cells, we say things like CAR T cells, may be curing some patients with these diseases. We tend to hedge. Maybe we don't have to hedge anymore. Maybe we don't have to say maybe. Should we say probably curing? Should we say things like likely curing? I actually think with this kind of data, we can say CAR T cells are curing at least some of these patients with these awful relapse refractory leukemias and lymphomas and have really absolutely dramatically impacted the field and impacted these patients' lives. And finally, let me just summarize by using the Philadelphia Phillies as a backdrop for where are we now with CAR T cells for cancer. I showed you some data applying them to ALL. That was a home run. ALL is way out in the bleachers. It was incredibly successful in the first FDA-approved product. But using it for non-Hodgkin's lymphoma is way out in the outfield as well. I didn't show you this data, but we've treated CLL with very similar results to lymphoma. That's certainly a double or triple out in the outfield, as is multiple myeloma. Again, I didn't show you these data, but it's been very successful. But excitingly, what I think is important is what's on deck. We have a really exciting program using CAR T cells for AML, but what you're going to hear about now is the next thing on deck, and that's using CAR T cells for autoimmune diseases. Many of these other targets and malignancies are still in the dugout, waiting to get into the game, waiting to see how they're going to perform. And even more exciting, I think, are those sitting in the stands to be named later, those kids looking on, thinking they just want to get a chance to be on the team. I think we're going to learn what these things to be named later are over the next several years. That's really going to have a dramatic impact on the field. And with that, I'd like to turn this over now to Dr. Merkel, really to explain how this is going to be applied and why it should be applied in rheumatologic diseases. So, Peter, it's all yours.
Thank you, David. That was, as always, outstanding and precisely on time. Well, hello again, everybody. I'm going to talk to you about the rational use of cell therapy to treat systemic autoimmune rheumatic diseases in general, with some specific ideas. But, how did I get to this point? I'm a big proponent of studying cell therapies and seeing whether or not these are going to be transformative for our patients. But it didn't happen overnight. There's been a lot of excitement in the last few years, appropriately, given the data that's out there. They're very limited data. But when I was first thinking about this was actually quite a number of years ago. I talked to Carl June and his group at Penn. We weren't quite ready because of the cost and expense and the idea and some of the toxicities that had been that David explained. But as things got better and better, and we got more excited, even before some of the data coming out of Germany, we were beginning to think about this for our diseases. And I began to partner with oncology and learn from them about what it means. And that was really crucial to really understand it, understand the safety, the efficacy, and what this involves. And this has led me to the end of the road where we're really talking about using this for autoimmune disease. And I think it's a journey until when you first hear about it, it sounds like this crazy treatment. And then we realize, actually, no, there's a safe way to study this. There's an appropriate way to study this. And that's what I think we're at now. So why autoimmune diseases? There are a lot of them. There are 80. This is actually, it really depends on how you count. Some would argue there are several hundred different autoimmune diseases. 1 in 10 people have an autoimmune disease of some sort. There are 50 million Americans potentially living with autoimmune disease. And of course, America is only 4% of the world's population. So there are zillions of patients around the world with autoimmune diseases. They're incredibly burdensome for patients. Compare that to non-Hodgkin's lymphoma, which is bad and is also important, but there are many more patients with autoimmune disease than any of the individual forms of cancer that David described. There's a big unmet need. We have not cured our diseases. So why are we targeting B cells? That's the first approach. I would like to make it very clear in my presentation. Cell therapy, right now we're at CAR T19, which is about B cells. We're already thinking about targeting other types of cells and other type of immune targets. But why B cells? Well, our diseases have lots of autoantibodies, and B cells are considered central to the pathophysiology of autoimmune diseases. And studies of B cell targeted therapies, especially rituximab, but not only rituximab, provide a scientific rationale and demonstrate the need, but also the unmet need. So we have a sense of what we can do. We also think there's ways to study the disease that we didn't even have before. We think we can get to an approvable drug. I want to quickly just introduce the idea that we've been talking about for several years in rheumatology was, where does autoimmunity begin? Is it before patients are clinically active? We're not going to use these drugs yet for those patients, but you can imagine that some of these drugs could go very early in the disease course once we recognize autoimmunity is present. We need to prove it. We need to get there slowly. But take a disease like type 1 diabetes, where we can actually identify children or adults at high risk for type 1 diabetes. What if we could turn that system off right now? But as we give CAR T therapy and cell therapies, we think about the B cells, we then give the CARs, and then we try to basically wipe out the bad B cells and have the patient repopulate themselves with naive B cells that are no longer pathogenic. That's the simplified view, but that's really the goal. So we were thinking about this, several of us, years before Dr. Schett's groundbreaking work, but when he published this one case of a patient with really bad lupus who had tried everything, and this woman who is still doing very well several years later has been was able to get off of her therapy and was able to be off glucocorticoids and off immunosuppressive therapy after having pretty bad renal disease and other things, was really remarkable. And he followed this up with another series of patients in this report and many reports that have followed that show both clinical autoimmunity is going away and sort of laboratory autoimmunity, that immunomarkers are also changing. So there's a change in the milieu of what we're doing. So we think that with those data and others, that CAR-T19 has the potential to be transformative for patients with B-cell driven autoimmune diseases, but we need to prove it. Why do we think this is the case? Because there's an unmet need. We know that their tissue-resident B cells are central to pathogenesis of multiple autoimmune diseases and are resistant to depletion. So we know that we're not getting all the patients, all the B cells with rituximab, for example. We know that cell therapy has the potential to be unencumbered to inflamed tissues. So one of the issues with rituximab or obinutuzumab is, are we getting to all the tissues? We know the blood B cells are gone, but are we getting rid of the body's B cells? And the answer is almost certainly no. Can we do a more complete tissue-based repletion with CAR-T19? And can we have a durable response? And that's the other flip side, is treatment and wiping out is great, but does it last? And finally, we think there could be an immune reset. We thought that perhaps rituximab, but that was not the case, although it's done very well for some of our diseases. But can we actually get to the point where the immune system is reset, is no longer pathogenic for the patient, and they no longer require lifelong immunosuppression? That would be absolutely remarkable. And so let's just this is a little bit what David talked about as well, but I really want to review the immunology here, because what we target really matters. So just to remind you that what we're really talking about, the differentiating factors for all of these different acronyms, all these different abbreviations on our targets, are the plasmablasts and the plasma cells, because this is the evolution and the progression of B-cell maturation, and it does make a difference. So for example, CD20, anti-CD20, rituximab, obinutuzumab is excellent, but not perfect. It hits this lineage, but it does not really hit the plasmablasts. It stops just short of them, or maybe get a little bit of early ones, and that makes a difference, as opposed to CD19, which is our CAR-T19. This goes all the way to plasmablasts and early plasma cells, but perhaps not the late, long-lived plasma cells, the memory plasma cells. All of this matters, because what you end up with matters. Do you still have vaccine response? Do you have to be revaccinated? And have you gotten rid of all of the pathogenic B cells and plasma cells that you need to do in order to cure a patient? Similarly, BCMA and CD38, these are two other targets, B cell antigen surface expression targets that are not only the target for some cell therapies, but also for bispecific monoclonal antibodies, and are being tested in lupus and other diseases. Very interesting, but look what it does. It really does something different. These things go for very late stage. As a matter of fact, you wouldn't get the early lineage, so sometimes you combine them with an earlier CD19 plus a BCMA. But careful what you wish for. You'll be wiping out plasma cells. Will you have recovery of those? Will you have to repopulate? So these are very powerful agents, and we need to do this in a careful fashion. David talked to you about how these are constructed. They're really actually unbelievable technology, but the technology matters. So what you can see is these are all different cell therapy products that are against CD19, but there are some subtle differences, and maybe not so subtle, in the hinge region, the transmembrane, and you'll hear about the co-stimulation molecules. What we don't know is how important those differences are. They do make a difference between how quickly the cells expand, how quickly they last, and we'll learn from comparative effectiveness about these things. But you need to think a little bit about this. Right now, we think they're pretty similar, but we will see if they're similar for oncology. They haven't been perfectly similar. Will they also be somewhat different for autoimmune disease? And similarly, you can see, especially in the co-stimulatory molecules. And you can see here that there are now CAR cells being developed against BCMA and CD19 and BCMA, and they're different with other ways of creating them. So we just need to be careful that we don't put all of this into one basket. There are different. So why are we thinking about our diseases? Well, I think here I list several of our diseases that we're looking at for treatment with CAR T, and the rationale, the unmet need, and the biomarkers. Let's go through these. For lupus nephritis, it's gotten the most attention because, to be honest, the results have been dramatic. I'm actually not going to show you a lot of results today of work that's not ours. Suffice to say, there has been remarkable interest in lupus nephritis because of the relatively small number of patients that have been reported who have been able to come off immunosuppression or mostly come off and had either reversal or halt of their nephritis. That has not been the case for all patients reported, and that's important to remember. And the sickest of the patients haven't necessarily been tested yet. But we're very excited about this. Why? Because we know that belimumab and obinutuzumab, which affect B cells, are effective in this disease, as is rituximab in certain settings. Obinutuzumab, those results I think are coming out soon for the phase 3 trial. Very encouraging about more denser B cell depletion. However, even with these treatments, we're not getting cures. We're not getting even complete responses in many patients. And that's simply not satisfactory. That is unacceptable to have continued active kidney disease. We have biomarkers. We can measure this disease. Of all parts of lupus, nephritis may be, I wouldn't say easiest because nothing's easy about studying lupus, but it is the clearest. We have a proteinuria as a marker. We can do repeat biopsies to look for immunologic response. We have biomarkers and immunologic responses, which are interesting, although not definitive. And there's a path to established endpoints, meaning the FDA, the EMA, and other international regulatory bodies have approved drugs based on certain parameters. And so we can look, that's important because we want to get these drugs into patients if they're effective. So for lupus nephritis, there's a big unmet need, there's a rationale. For ANCA-associated vasculitis, which I've done a lot of work in, much more than I've done in lupus, although I'm doing that now. One of the things we know is rituximab is approved for treatment and it's extremely effective. We also know that obinutuzumab appears effective as well. And we have still unmet needs for two reasons. One is there's some patients who just don't stay in remission with rituximab and have relapses. And the second reason is, do we really want to put people on rituximab for 20 plus years sometimes? Chronic B cell depletion, immunoglobulin deficiency, et cetera, has its risks. And so what if there's a way to actually reset the immune system so we wouldn't have to do that? So I would argue that ANCA-associated vasculitis is the condition within rheumatology for which there is the most rationale for B cell depletion because of its great success, but perhaps less need because actually our patients are doing pretty well once they're in treatment, but it's a different type of need. Additionally, we now have 2 drugs approved, this plus avacopan, and so there's a pathway to approval. I can't tell you how important that is. Rheumatoid arthritis, by far much more common than all of these other diseases put together, 1% of the adult population. Again, B cell depletion is FDA approved, has been shown to work in a subset of patients. And I think the subset idea we have to keep in mind because I think if CAR T and cell therapies are going to work for our diseases, and I hope they work, and I hope they work incredibly well, they're probably not going to work for everybody, and there may be subsets that they work better in, and we need to identify those. But we have lots of drugs for rheumatoid arthritis, of which B cell depletion is one approach. But not everybody actually responds. There's still plenty of unmet need in rheumatoid arthritis. And again, this idea, do you want lifelong immunosuppression? And again, we have very clear pathways. We have outcome measures. We have outcomes that regulatory bodies are happy to help work with us on to get drugs approved. Then we go into the diseases where there's been less defined path. Systemic sclerosis, scleroderma, I've been studying that also for 30 years. We have data, and I'll show you a little bit of that, of rituximab and B cell depletion playing a role in the pathogenesis and perhaps treatment of systemic sclerosis scleroderma. We don't have good treatments. The unmet need is huge for all aspects of scleroderma. We do have biomarkers and outcomes we think are effective, and we have some defined path. There have been 2 drugs approved. So there's an approach that we can work with the agencies to get this done if we do CAR T. And then the myositis, I should really combine these now. The idiopathic inflammatory myopathy, our new term that we like to group everything together, including necrotizing myopathy. But separate these because they have, at least in Dermato, have led to approval as well. There has been mixed data on the use of B cell depletion. When reanalyzed, there's really a sense that this is probably something that we should certainly look at in myositis. There are better outcome measures than there have been ever, and we think there is a pathway to regulatory approval. Maybe more difficult than for RA, but that shouldn't stop us. And there's a lot of unmet need in this disease, both the muscle disease and, of course, the lung disease and other aspects of it. So, oh, I'm clicking the wrong thing. Rituximab, I just want to go back, and I'm going to go quickly because many people feel that, wow, this CAR T is pretty dramatic that we're doing all this. But I would remind you that when we first did rituximab, this was considered radical, and some people thought we were reckless when we did this work, which I do not think so. We did our RAVE study. We had almost 200 patients. That was based on 10 patients with severe refractory AAV treated at one center at the Mayo. That's it. Then we planned this trial because there was unmet need and there was the potential. So it's not that different from we have a relatively small number of patients getting CAR T, and now we're looking at it for different diseases. But we strongly protected patients. We had a careful approach at expert centers that knew what they were doing, and that's key. We were uncertain about the ability to recruit, but you know what? I was at Boston University at the time before I moved to Penn. We got 41 patients into this trial. Patients with rare autoimmune diseases are highly engaged in our work and are happy to participate if they feel they can help others in the future. And that's the reason to do this. We proved it worked and it changed the field. The reason I'm saying that is it was kind of out there at the beginning, just as this is. Now, there have been other things that are out there that didn't work, but we need to go carefully forward. And this was of course, our paper that got it approved. And this is our second paper about the long-term follow-up. And I want to show you this because, yeah, it works well, but look what happens after one course. Just as with cyclophosphamide, you see steady relapse. And I can show you lots of different data from different trials, including one we published last year on repeat treatment. When you stop therapies, or even if you continue, relapses continue. So we have more ways to go. So it was radical, but now it's the standard of care, but we can do better. Similarly, for systemic sclerosis, scleroderma, we did a very early study when I was in Boston on the use of rituximab, and we found some very interesting markers that changed. I won't go through this in detail, but there are some reasons to believe that the skin disease was changing with B-cell depletion. This is a study from Japan from a few years ago that really opened our eyes. This was treatment with rituximab, and what you can see is there was a substantial difference there was a reduction in skin score, which is, how thick you are, how involvement is there in the skin. There was a substantial reduction, which we hadn't seen in previous studies, compared to placebo, and there was an improvement in lung function. These have not yet been replicated, but quite interesting that B-cell depletion should work. And every subgroup was in the right direction. So there are varying levels of support also for lupus, and I think there's going to be much stronger support when we get the obinutuzumab data published, and for myositis. So I want to show through all of it. You know this well as rheumatologists. You've seen this. There are different levels of support for B-cell depletion. What about our current options? Well, obinutuzumab is a denser B-cell depletion, and it might be the right answer for some patients, but it's not going to be transformative in terms of turning off the immune system. It's unlikely to cure, but it may be good enough, and so we have to keep that in mind. But CD20 depletion is not the same as CD19 depletion, so we need to think about those long-lived plasma cells, et cetera. And the pandemic did a lot of things, but one of the things that made us very much reconsider the impact on vaccine responses to our therapies and the impact of chronic B-cell depletion. Our patients were not doing great to our chronic B-cell depletion. They're still an issue. And in general, we should be concerned about chronic B-cell depletion. It is a risk. It may be better than the disease for sure, and better than being glucocorticoids, but it is a risk. And if we can figure out a way to not have patients B-cell deplete and not be hypoimmunoglobulemic, obviously that's better. So I think we could do better. And here is some interesting data published by Georg Schett's group. Again, they've done some foundational work in this area. These are based on inguinal lymph node biopsies. And what you can see here is that in the first row on top, before CAR therapy, you can see the CD19 and CD20, they're all lit up. There's lots of them in the B cells and in the lymph nodes. The second row is after CAR therapy. And what you can see is there's not only are there no cells, there's no architecture left in the lymph node. It has stopped being that kind of lymph node. So it's really changed things. On the bottom row, this is after rituximab, also inguinal lymph nodes. There are fewer cells, but there's still some B cells. And so this whole concept is let's go better and deeper and longer in B cell depletion. So this would be a quantum change. This would be a definitely different approach to therapy for something we've never seen before. But there are some precedents for this, and including in autoimmune disease. I'm just going to give you a sense of something else. In amyloidosis, AL amyloidosis was uniformly fatal. High-dose melphalan with anti-plasma cell therapy was used, and it was then there was a reluctance for its use in AL amyloidosis because these patients were quite sick and they had the potential to die with this. They had cardiac disease, etc. Boston University and then other centers, not me, my colleagues actually, began using high-dose melphalan and autologous stem cell transplant. And they did this in a multidisciplinary team, which is why I want to bring this up, because I can tell you without the combination of rheumatologists and nephrologists and oncologists and cardiologists, this would not have done well. But because they worked together and took care of these patients who were sick, they were able to show that this therapy was remarkably effective and changed the disease. And they published multiple papers looking at this in different settings, and it became the standard of care. Here is for example, on renal disease. I particularly like to showing this because my wife, who is downstairs, a nephrologist, part of the group, she's now leading the same ideas at Penn, was the lead on this as a nephrologist, and this changed how patients with kidney disease in AL amyloidosis were treated. They have some new treatments now, but that's 20 years later. And this was really radical. So the point is, with proper care, with treatments that appear to be edgy, if you do it right, select the patients properly, and number one focus is on patient safety, you can do this well. Similarly, high-dose chemotherapy and autologous stem cell for scleroderma. This is, there are two trials, one in Europe and one in the United States. And what that showed was that patients who got this high-dose chemotherapy with stem cell recovery, their own stem cells, did better from a survival standpoint than their scleroderma. This, however, has significant toxicities. There are some relapses. We are doing this at Penn, and we still welcome patients to be enrolled and to do this. And it's not, I wouldn't say it's standard, but it is an approach. This tells you again that substantial immune reset can make a difference. So what are the common features? Standard of care was unacceptable or unacceptably ineffective. The recognition by stakeholder groups, including patients, that we need to do better. There were good pilot data, sometimes not that much, but safety was the number one concern. You get a multidisciplinary team together and you can move it forward. I think that's the current state for CAR T in multiple diseases, in lupus, scleroderma, myositis, vasculitis, RA, inflammatory neurologic disease, including multiple sclerosis, myasthenia gravis, stiff person syndrome, and other autoimmune diseases. We can do better. So we need to actually work together. We'll talk about that in a moment. So I don't think it's reckless. I think it's cautiously forward thinking. This report from the German group was yet another one. This was last year. And what you can see is they had now 8 patients with lupus. They've treated quite a few others. And there have been some reports from China. There have been some other reports from around the world showing significant benefit in most, but not all patients. And I think that's what's important. Now, will this be able to be scaled up to more patients with maybe more severe renal disease or early in the course of disease? Let's do the experiments and find out. Can we determine who's going to respond and who's not going to respond? Let's do the experiments and find out. I don't think it's going to cure all of our patients, but what if it cures some of them? And what if it's very effective treatment? Similarly, but there's also the possibility it won't be for some diseases. This is myositis, good results, but again, very few patients. There's been some dramatic results in patients with synthetase syndrome and lung disease, ILD, but let's find out. And here's some patients with scleroderma, and you can see, again, benefit. So we need to make sure it can happen at other centers with other products, etc. So there are now several reports from different groups of the treatment. We have treated some patients at our center as well with these diseases. And this is mostly positive, some mixed results. The range in follow-up, though, is for greater than 3 years for that first patient, and she continues to do well in Germany, to 6 months. It remains experimental. One of the big questions is, will the disease come back? And if so, in what manner? And who should we treat and how should we treat them? But the early results are very promising for this approach to immune reset. But there are some other options. What we've been talking about today is autologous CAR T, meaning that the patient's own cells are taken out of their body, processed, and put back in as CAR Ts. But there's also the idea of allogeneic. So somebody else's CAR Ts from a healthy donor are taken, they're transformed to be CAR T, and they can then be infused. And the reason for that is you don't need leukapheresis, you don't need some of the prep for that, and you can just infuse them. The other advantage to this would be that you can be very selective about the cells you want. And another advantage is it could be much less costly because this is very costly. To what extent this will be accepted by patients and not immunologically active is still clear. It's been effective in some forms of cancer, and we can talk about it on the panel. I think the most exciting part, something that Dr. Porter and I are both working on at Penn, is the idea of in vivo. So this is, and I'll show you in a second, the idea you make the cells inside the person. You don't need to actually do lots of other manipulations, and through a variety of very advanced techniques, can you grow your own T cells that are going to cure you. So we talk about T cells because we really like to talk about T cells, but there are other cells. Gamma delta T cells are different from the T cells we're using for CAR T. NK cells. These all have advantages and potential disadvantages for use of cell-based therapy, and they're all being investigated for lupus and other diseases now. There's also the idea of combining the two or three. So we have a lot of different tools, perhaps too many right now, to study effectively. But we need to get a sense of what's going to work best. You heard about the viral approach to doing this. We're looking at also this LNP. We're very big onto mRNA at Penn, where, of course, our colleagues won the Nobel Prize for using RNA technology that led to the vaccines. Same approach, actually, with these liquid nanoparticles to potentially put in CAR-loading mRNA and create the CAR cells in a person. You get an infusion, you create your, and this actually transforms some of your cells into CARs. You don't need to get chemotherapy, you don't need other things. And you can redose, and you can put more than one thing in together. We're far from that yet for people, but not that far. And we're very interested in doing this. We're getting there soon, we think, to test. Similarly, there's CRISPR technology to even be more fancy and cut things up and make some differences. So again, different approaches, but we need to prove it. So just to remind you that these last two approaches I spoke about with the in vivo would be in the person themselves. So there are also these really cool, you have to have acronyms. They're very dizzying. So the alphabet soup is dizzying, but these are CARs with 2 A's, and this is antigen specific B cells. So for example, against the antigen for membranous nephritis or the antigen for myasthenia gravis. You could see where that would be effective if you know the antigen. And similarly for these other ones with T cell receptors, and then this super CAR, which combines 2 different ideas. So what are the concerns about? I showed you the excitement, and I think you've all seen the data where patients are, they're off therapy. And I would argue if you have a patient with lupus and she has nephritis, and in a year she's off immunosuppression and on low dose or no dose prednisone, and her proteinuria is as good or better than before, my God, that's fantastic. We'd love that for someone who had difficult disease. I'd like it even better if it lasts 5 years or 30 years. So what are the issues? Well, one of the issues is they may not work. And so we need to be accepting of that. What do we need? We need data. That's how you figure that out. The lymphodepletion. So Dr. Porter explained people need to get cyclophosphamide, fludarabine. That's the current regimen. That's significant lymphodepletion. We think there will be ways to reduce or eliminate that over time. In vivo would eliminate that approach. The cost, hundreds of thousands of dollars. That may actually be cost effective if you think about the cost of taking care of patients forever. But these costs are absolutely going to go down with technology, and there are some other approaches that will bring the cost down. Prolonged immunosuppression, chronic immunosuppression. Some of these things like the BCMA may lead to more chronic immunosuppression. We need to be careful, and we need to test And we'll see, it depends on the target cells. We may need to revaccinate. I don't think so for the current CAR-T19, but they may be for other ones. Secondary malignancies, we can talk about that at the panel. There have been a few reports. These are extremely low risk, but it's certainly bad if you're the patient. We need a little more research and longer term follow-up. Can we get there with bispecific antibodies? So the bispecific monoclonals you're going to be asked to enroll patients in these trials. They're very interesting. They may be very effective. Will they be as effective? As we will have to see. Let's do the experiments and compare. And finally, there's really too many companies doing too many. There are probably more slots in CAR-T trials right now for lupus nephritis than there are patients who can enroll. And this is a problem. The industry has to shake out. We need to work together to figure this out. We need to develop a rational approach that will lead to regulatory approval. And I think we'll get there. Fallout will occur, as I say. So to conclude, cell therapies offer great potential to transform treatment of various autoimmune diseases. The possibility of long-term drug-free remission, and I'm going to use the word cure. I like to use the word cure, as Dr. Porter said. That should be our goal. It's too important to not investigate. These approaches remain experimental. I think we need to care. There's a appropriate excitement, but we don't want, we don't want patients to be and physicians to have, therapeutic misconception. These are experimental studies. We need to apply the same scientific rigor to CAR-T as any other new treatment, although we may have different designs. And with that, I think I'm going to end. I think we're very, very excited about how this is going to work, but we need to test it out. And I think what we're doing now isn't what we're going to be doing in 10 years or 20 years, but hopefully this is the start of a really new way to treat autoimmune diseases. But I would be remiss if I didn't bring David Porter back in and to say, we do this in combination. I can't tell you how important it has been to learn from him about what these therapies are and what they're not, and how to treat this. It's not just the therapy, it's not just the toxicity, it's the machinery at the hospital, it's the immunology we teach each other. It's a lot of things that really make sense. And I think we're trying to break down these barriers and work together. David, do you want to speak to this as well?
Yeah, sure, Peter. That was wonderful, by the way. But I think we were talking before the session that the medicine part of this is almost straightforward. We are repurposing a drug for autoimmunity. This is a medication that we use, that there have now been over 42,000 people around the world treated with commercial CAR T cells for cancer. And I think we understand how to do that. What has been really complicated in our world is how do we apply this across disciplines? And this is just a cartoon to show you the way that we think of this traditional model. We have the cell therapy program, which started within my group. We are oncologists and bone marrow transplant physicians. And while we have colleagues who are rheumatologists, dermatologists, nephrologists, neurologists, we certainly don't do clinical trials necessarily together. I think what has been critical to success is the ability to really break down those walls. I'm sorry, I think we're both controlling the slides, but and we've had success with some support through our Colton Center for Autoimmunity at Penn, but this is really what is necessary to move this field forward and to be able to apply these therapies really across disciplines. Cell therapy is a modality, it is a specialty unto itself, and this has to be a collaborative process to be able to do these clinical trials and treat patients safely.
I would agree, and I think it's also really cool science, and we have been able to do. We're collecting cells. We're going to learn about the immunology. I think we've learned a lot about immunology after we figured out these new therapies. All our biologics, for example, are teaching us about the immunology of our diseases, so we're going to learn that as well. At Penn, we have a collaborative approach when we've treated now some patients with lupus and scleroderma with CAR-T therapy, and it is a continued give and take for choosing the right patients and setting up the protocol and getting it done. And if a patient, they are seen by both sets of physicians. We do this as a group to collect, including our nephrology colleagues together with us, and we choose the right patients to move forward. And it's gone very well. This is a list of things that we're working on, and some of the people involved. And I don't know if there's more to say here, David, but we're open to thinking about this. We think this is, as I said, the potential for cell-based therapies to be transformative, even curative for treatment of some patients with some forms of autoimmune disease. You can hear the hedge. The hedge is nothing's perfect, but we think I would be surprised if some form of cell-based therapy, whether CAR-T19 or in vivo or others or all of them, isn't incredibly helpful for some patients with some forms of autoimmune disease. I would be surprised if that was not the case. I'd be delighted if it's for a lot of patients, but we'll have to see. And then, I don't know if you want to add to that, David. But I was going to say, so why believe us? Well, we're not alone in thinking about this. This is from a couple of months ago in The Economist. I was flipping through my issue and I said, I actually know about this. And there was really great excitement about this, because there's really so much going on in this field that we're going to learn. So I think the world is watching as physicians who deal with autoimmune diseases are trying to address this issue. And I think we're happy to take questions. David, do you want to explain this modified skyline of Philadelphia?
No, it is what it is. That is indeed, for those of you who aren't local, the modified skyline of Philadelphia. And if you look closely, you can see all the science.
Yeah, and if you look very closely, or if you looked at it tonight, you'll see that it will be lit up in green. We have a whole coordination of the buildings, and that's because we, of course, are all rooting for the Eagles to win tomorrow.
Purple today, green tonight.
Exactly.
Those are fighting words.
I try to speak the truth as I know it. I've data to support that statement.
Well, thank you both. That was an incredible, amazing run through of a very complex topic that is really top of mind for all of us across both specialties. And I just want to start by saying, a bunch of questions coming through, and I'm going to kind of group them. And the first is, how do you know which car to choose? I mean, it's much like buying your first car. You want to make sure it's the right car, it's the best car, it's the one that's likely to be durable. How close are we to being able to answer that? I know oncology is 2 decades ahead, but we know, Peter, that in our space, we're not even sure what the exact target and the best marker is. So how close are we to choosing the best car?
Well, maybe we can each answer. I would say right now, the targets, almost all of the targets are the same, which is a CAR T19. So right now it's the CD19 positive cells. So that is the current most common target. As I mentioned, NK cells and gamma delta and other cells are coming into play, and probably antigen specific. They're coming into play. But right now, it's all the same four-wheel drive SUV. The question, of course, which brand do you use? And that's really, and the difference matters. But maybe. They probably are fairly similar in their approaches. The design of the trials are somewhat different. The dosing may be different. The ability for the cells to expand inside the person may differ. The ability for them to persist or not persist, depending on what you want, will be different. And then we start adding these extras you can get at the dealership with T-cell engagers and other things that will help push things one way or the other. I think the answer is data. And to do it right. But I think this was, and I think that this is the first generation. This is like the third or fourth generation or fifth generation for CAR T, but it's the first round for autoimmunity. And I think we're going to see different things in 5 and 10 years. But David, you faced, and you continue to face the same issues in oncology. What are the lessons learned there for choosing?
Yeah, I think it's a really good question. And I think, Peter highlighted this in his talk. You got to do the study. You have to do the experiment and learn. But there are differences in the different CAR products. And again, if we just take the ones that are now commercially available, there are really three major CAR T cells for B cell malignancies, and they all have the same targeting antibody, and they differ in the co-stimulatory domain, and that probably does matter. So, for instance, products that use a 4-1BB co-stimulatory domain, which is the one of the ones we developed here, that's the one known as Tisa-cel, or Lysa-cel is the other one. They tend to have less of a lower toxicity profile with a 4-1BB co-stimulation. The toxicity tends to be more predictable, slower in onset, and less intense. Those with a CD28 co-stimulatory domain, one under the name of Yescarta or Axicel, which is the Kite product, tends to have more toxicity, more rapid in onset. However, those with a CD28 co-stimulatory domain tend not to persist as long, whereas those with a 4-1BB co-stimulatory domain have long-term persistence. I bring that up because in oncology, that's important. In ALL, we know that persistence of the CAR T cell is really important. It's a quality attribute. And therefore, in ALL, people favor a CAR T cell like Tisa-cel with a 4-1BB co-stimulatory domain. In lymphoma, or diseases, mostly lymphoma, maybe multiple myeloma, it's not so clear that you need persistence. And so the other products may be just as good and maybe even better. In rheumatologic diseases, I don't think we know yet, but the general hypothesis is you may not need long-term persistence. And therefore, and maybe long-term persistence gives you long-term B-cell aplasia, and persistence may not be what you're looking for. Again, as Peter showed with some of the data of autologous transplant we see it in myeloma, but in amyloid, you get this immune reset. You just need really deep B-cell depletion, then the B-cell repertoire recovers normally for reasons, I must say, I don't understand. So in autoimmune disease, if that is indeed the case, maybe the CAR with a CD28 co-stimulatory domain, which has very potent T-cell depletion, but isn't expected to persist, may actually be a good thing. Again, it's a very long answer, but you do the experiment.
I want to add 2 points, which I think we didn't get to. One is that this was described to me actually by Nash Hussain, who's an oncologist, works with David and me, who's terrific, that the 4-1BB is a Prius, it's reliable, it moves forward, and that the CD28 is a Tesla, and it zooms up, and then, it goes down. So there really are, it was a car analogy. They are different. The other thing is the toxicity we see in oncology is because if you've got B-cell lymphoma, you've got a lot of B-cell burden. You've got a lot of tumor out there. In rheumatologic disease, we think there's much smaller volume. There's just less target tissue. So there's fewer cytokines to be expressed if you kill all the cells. And so we think the toxicity, not surprisingly, on the cases that have been out there reported, there's toxicity, but there's not to the degree that you would expect if they had B-cell lymphoma. There's just not as often, and it's not as severe, and I think David's nodding, and that's because the burden is lower. Does that mean we can do some things differently? I think almost certainly, but I think this idea of, do you want persistence or not? And there are some companies that we're working with who are thinking about, maybe you do want a little persistence for some things. Maybe you don't want to go. So, one described it to me as a chill, not kill approach, where you go slowly. Now, whether that's appropriate, I don't know, but I would say it's a happy problem if we have more than one effective approach and we have to compare them.
So that brings up the idea of, can we possibly have varying lymphodepletion regimens to make this more rheumatology friendly, more outpatient friendly, less toxicity, especially ICANS, I think, and CRS scares many of us in rheumatology, because we don't really want to have to deal with that depending on the disease we're dealing with to begin with. So is there a possibility that we can do this a tad bit differently as a result?
Let me answer, and then I'll turn it more over to David, because he will tell you. I think we don't want any patient to suffer these things, but almost always these are reversible and are addressable. And David has taught me that we can deal with these. And but I think we will end up with regimens that less immunodepletion. The basic strategy is we should go with what we know, and what we know is cyclophosphamide and fludarabine in the regimen. And if we show it's effective, then we could start looking at less and less lymphodepletion. But if we don't do that and we fail to show benefit, is it because of lymphodepletion didn't happen? And so we need to first show benefit and then try to find other ways. And that's why in vivo and other things are attractive, Grace, because you don't have to do the lymphodepletion. I will tell you that these are toxic, but so is giving cyclophosphamide a few times to a patient with lupus over time. So is some of the stuff we do, some of the glucocorticoids we give. I'm not rationalizing, I'm just saying, don't underestimate what we've done to some patients. And I think oncologists have a different view of the toxicity, and they're not because they're cavalier, but because they've been able to handle it. David?
Yeah. It's right. It's a little bit what you're used to. Chemotherapy still has a terrible connotation, which maybe isn't always deserved. In oncology, there have been good studies, and lymphodepletion is important, if not critical. There were studies using Cytoxan as a single agent, and if you add fludarabine to that, the CAR T cells work much better. I mean, the experiments were done, and we know in that setting, lymphodepletion is important. And I would venture that a lot of these oncology patients are probably more immunodeficient, immunosuppressed at baseline from their prior therapies, maybe than patients with autoimmune diseases, though patients with autoimmune disease have been heavily pretreated, at least with immune suppressants. So I think lymphodepletion is important. It is indeed, the side effects are manageable. There is a lot of interest in trying to figure out what the limit is. There are some patients who come in immunosuppressed because of their prior therapy. They have low T cell numbers. They maybe don't need it or don't need as much. But I think that it is going to be needed. The in vivo approaches will be designed without lymphodepletion because you don't want to kill the T cells that you're trying to essentially genetically modify. If that works, that's great. It will be done without lymphodepletion. Whether it can be done without lymphodepletion, will those cells still expand if you haven't lymphodepleted the non-genetically modified cells or not, I think remains to be determined. So it is understood this is a major issue. I think to the oncologists who do this, the lymphodepletion is generally thought of as mild and tolerable.
And I would remind us that, I mean, I think and that's why actually we've had our patients are consented by both oncologists and rheumatologists so they can actually discuss these things. I would remind us all, and I'm not, again, that when the NIH under Tony Fauci's direction started using cyclophosphamide for vasculitis, it was pretty radical. And then it became standard of care for 40 years. And so, and then we started using it for lupus, and now we're getting away. But, you're a 35-year-old woman with lupus who has already seen cyclophosphamide a few times, and you haven't had the children you want. That's a very different decision than if you're younger or if you're a different stage in life. So, as is always the case, but I think we can do it. And David, you might want to talk to, because your group is working very hard on outpatient therapy for CAR T.
Yeah. And I think just to go back to that, the other kind of buzzword, whatever, in the CAR T cells are designed as a one and done therapy. And things like long-term toxicity and fertility have to do with repetitive cumulative dosing. And with the doses that are used in the one and done approach, it really seems to be more manageable. But it is true. We have been doing this since we started doing it over 14 years ago in an outpatient setting. The toxicity tends to be predictable with most of these products and manageable. It doesn't mean that some patients don't get admitted, but in our experience with the right product and identifying the right patients, about 2/3 can be managed through their whole episode of care as an outpatient. And that dramatically improves access. Patients are much more willing to think about it when they don't have to come into the hospital for 1 or 2 weeks. You have to have system set up where you can monitor them. You need the infrastructure to monitor them, to have cell therapy expertise 24/7 by phone and in an emergency room and also in an inpatient unit. But absolutely, this can be done as an outpatient, and I think that is the wave of the future.
They're
Especially
done
as you
as
mentioned with autoimmune disease and the low target burden, we expect lower toxicity. I think that's even more conducive to outpatient management.
they're done as inpatients now because these are early experiments and the
Right.
products haven't been tested in this population. And also in different parts of the world, there's a different tendency to put patients in hospitals for experiments and different
Right.
things. So we have to sort of think about that. On the other hand, it may not be that everybody can't necessarily administer CAR T. You really want to, you got to know what you're doing. Just as you got to know what you're doing as rheumatologists, we don't think everybody should use our drugs. You got to know what you're doing.
Mm-hmm. Absolutely. We'll have to have you come back for round 2 to actually talk through that component because otherwise we don't get enough patients into this sort of new and innovative way of thinking. And we're going to have to wrap this up. I am really sort of very saddened to have to say our session has come to an end. But we'll shoot some questions. There was a question about the underlying biology of ICANS, the infection rate. But if we'll do that offline, and then we can just add that to our chat references for everyone. So a huge thank you to both of our speakers for walking us through complex biology, oncology, rheumatology, and ultimately ending with collaborative care, which is always where I like to land, because we do better when we work together, much, much better. So thank you,
Thank you for
Peter
having
and
us.
David. You've gone from Dr. Merkel and Dr. Porter. But thank you for this wonderful time. And Dr. Reddy is going to now take us into our break and tell us when we should come back. So thank you all for staying. We'll be back with the second part of our program soon. Priya, take us away.
Thank you.
Thanks so
Thanks for
much
having
to
us.
our speakers.